హలో మీ హలో
వచ్చింది కదా ఇప్పుడు వచ్చింది. అదే కొద్దిగా డిలే ఉంది.
1 minute
అదే ఎందుకు అర్థం కావట్లేదు. మనోడు డిలే, మనోడు అక్కడ ఏదైనా లేటెన్సీ మాట్లాడింది కదా. హలో సార్, ఇది ఆడియో వస్తుంది కానీ లేటెన్సీ ఎంత పెట్టారు సార్? లేటెన్సీ లో లేటెన్సీ అయినా డిలే చాలా వస్తుంది. సరే సార్, ఓకే సరే సరే ఓకే. పర్లేదు, ఏం చేస్తాం. ఇప్పుడు మనం ఇక్కడ చూస్తే మనకు అనిపిస్తుంది. ఇప్పుడు డైరెక్ట్ చూసే వాళ్ళకి ప్రాబ్లం లేదు. వీడియో లేదు అది కొంచెం ఉంటాయి. చూసే వాళ్ళకి ఏమి ఇష్యూ ఉండదు, మనకి మాత్రమే అట్లా అనిపిస్తుంది. ఇంకో లేదు లేదు కెమెరా ఓకే, కెమెరా ఉన్నాయి. సరే, ఇంటర్నల్ ఉన్నాయి.
Good morning, everyone, and warm welcome to the International Symposium cum Workshop on Innovations in Translational Therapy and Targeted Drug Delivery, ITTD-2025, here at BITS Pilani, Hyderabad campus. It's truly a proud moment for us to host this 4-day gathering of distinguished scientists, academicians, clinicians, and industry leaders from India and across the globe. Over the next few days, we will explore the frontiers of translational medicine, from nanocarriers and biomaterials to immunotherapy, antibody-drug conjugates, and antimicrobial peptides, with a shared mission of bridging the gap between bench to bedside research. This symposium is a part of prestigious SPARC initiative, the scheme for promotion of academic and research collaboration supported by the Ministry of Education, Government of India. It brings together collaborators from Nanyang Technological University, Singapore, and La Trobe University, Australia, alongside BITS Pilani, creating a vibrant platform for international research partnerships. We are deeply honored to have with us today some of the finest minds in science and industry with us. Our guest of honor, Padma Bhushan awardee 2022, Dr. Krishna has been a true visionary in vaccine innovation. We are truly honored to welcome him with us today. Along with him, we have Professor Jimmy Shia from NTU Singapore and Mr. Vijay Gopal Chakraborty from UGA US LLC, who will share their cutting-edge work in biosensing and industry-driven drug development. Over the course of ITTD 2025, participants will not benefit from plenary and invited lectures, but also from hands-on workshops covering nanoparticle characterization, microscopic techniques, in vitro and in vivo characterizations. As we begin, I sincerely thank our director, Professor Soumya Mukherjee, our dean, administration, Professor Yogeshwari ma'am, faculty colleagues, student volunteers, and our international collaborators for making this event possible. I also thank our industry sponsors and American Chemical Society for supporting awards for oral and poster presentations. Therefore, on behalf of organizing committee, I welcome you all once again. Let us together make ITTD 2025 a memorable and impactful event that sparks new ideas, collaborations, innovations for the future healthcare. Thank you all.
We would now like to invite our guest of honor, Dr. Krishna on stage. May I also request our dignitaries, Professor Soumya Mukherjee, Director of BITS Pilani Hyderabad Campus, Professor Aarti Dhar, HOD, Department of Pharmacy, Professor Sonak Roy, SPARC Campus Coordinator and Dean GCIR, BITS Pilani Hyderabad Campus, Professor Jimmy Shia, Professor of School of Chemical and Biomedical Engineering, NTU, Singapore. I would also request Professor Yogeshwari, Dean of Administration, BITS Pilani, Hyderabad, and Professor Vamsi Krishna, Dean of AGSRD, to come on stage. I would also request Professor Mohan Rao to join us on stage.
To mark the auspicious beginning of the symposium, may I now request our guest of honor, Dr. Krishna Ella, Director, Deans, and the distinguished guests to join us for the ceremonial lighting of the lamp.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रान्विता या वीणां वरदण्डमण्डितकरा या
Thank you to all the dignitaries. May this event be enlightened with knowledge and innovation. I now invite all the dignitaries for the unveiling of the ITTD-2025 abstract book, showcasing the rich collection of research contributions to the symposium.
Thank you all. Now I invite our respected director, Professor Soumya Mukherjee, to share a few valuable words for the delegates and attendees.
Good morning, Dr. Ella, Professor Sia, Professor Mohan Rao, all my colleagues here and students. I do not know, first let me confess, I do not know anything about drug development or drug delivery. I just know that it is necessary. And one thing I know is that my daughter took that Pfizer medicine, whatever, she contracted COVID twice after that. So I'm not particularly happy with that, whatever. I did not get the Covaxin, unfortunately, sir, but we had to take the Covishield, which still works better. But most importantly, one of the things is which I think Professor Shia is going to highlight is what we sense we cannot control. So it is going to be a combination of a detection plus drug delivery, which is indeed going to be very, very interesting for even me, who is a sensors person. But these are the days of AI. Everyone says AI, ML, AI, and all sorts of other combinations of the same letters. The question is, this is what Dr. Mashelkar said long time ago in some meeting when Lakshman was hit by a Shakti shell and Jambuban asked Hanuman to get that Vishaldakarani tree. He got the whole mountain. Now, the moral of the story is that yes, we can get the whole mountains, but it is better to get the specific leaves, or in this case, nowadays it's good to get the tree, but it is even better if you can identify the active molecules. And not only that, with AI tool sets and things like that, it is possible today to develop specific drug molecules which can be of use, and that cycle of the initial development has now probably reduced. I can only say probably because I'm not an expert in this area, but still the requirement of such use of such targets on animal models and then human trials and all that still do remain. I do not know. We might be able to reduce the cycle using some various technologies, but still there is a significant amount of work to be done in that respect. And I believe Bharat Biotech, being one of the pioneers in the country, is indeed going to lead the way towards that development. It is, and in today's world, it is becoming important day by day to create our own niche, which no one literally can touch, no other country can touch. And there are industries in India which I can proudly say who have created the niche, and I'm extremely honored to be in the presence of Professor Dr. Ella, who has led such an effort. And if you have not read the history background story of how Bharat Biotech came up, then it is something indeed worth reading. Now, again, I'd like to emphasize that this symposium, conference, meeting, SPARC led meeting is going to be a brilliant one. It is going to be one with exchange of ideas. It is one where people are going to learn from each other and probably get rid of the world's greatest scourges, some of which are coming back in spite of our having thought that we have gotten rid of them, some of them newly emerging from zoonotic species, and some of them actually being developed, sort of quote unquote, in terms of biological warfare in various places. We should not use the word, but that unfortunately is true. And we have to protect ourselves against that. We have to have our vaccines, we have to have our medicines. We have to have personalized medicine for a variety of things. The old Ayurveds, they would study a person, they'll ask them all sorts of questions, almost for 2 hours they'll ask them questions. Many of them may or may not have contributed to the medicine or to the diagnosis or to what they did, but they gleaned some information from it and made personalized medicine for each of their patients. And probably it is time that we do look back at Charak Samhita and other ancient textbooks to understand what they were trying to understand and use the tools of AI to develop personalized medicine, which, while leaving the least impact on the human body, is going to create the maximum impact on the disease or the abnormality or whatever is happening to the person. So I hope that such meetings continue in the future, where we do have an amalgamation of the ancient knowledge and the new techniques, and put their hands together in developing newer and newer therapies for the human race as a whole. Now with that, I thank you very much. Thank you for participating, and have a fantastic time in the search of knowledge through the symposium. Thank you.
Thank you, Professor Mukherjee, for your inspiring words. I would now request Professor Yogeshwari to come and say a few words.
Very warm good morning to everyone present here. Dr. Krishna Ella, this is not the first time he's visiting the campus, and today he's Padma Bhushan awardee, which is a proud moment for all of us. I remember 10 years ago when Dr. Ella came to the campus, we were smaller than this. I think we did not have the auditorium, I believe. It was just, the auditorium was just inaugurated. And I still remember when he was addressing the students, he did talk about entrepreneurship, which was too early for our campus because we was just thinking about entrepreneurship, and he gave a lot of ideas. I would say his talk was 30 minutes, but he gave about 20, 30 ideas for students to develop. Thank you very much, Dr. Ella, for accepting our invitation and making this event a grand beginning. I also appreciate Professor Jimmy, Professor Swati Biswas and her collaborators for making the second edition of this international conference and workshop for a 4-day event, which is definitely a hectic planning. And I really congratulate Professor Swati for putting up all the efforts to bring in the second edition at the right time. I think last July we had the first edition. And I also have the responsibility of being a dean administration. I would like to tell the dignitaries as well as the participants that we are currently marching toward, this campus is marching towards its 18th year. This year we celebrated our 18th foundation in April 2025. And we have grown in numbers, we have grown in the facilities, and today we stand almost with respect to the research outcome or with whatever programs we offer, we stand in par with our mother campus, BITS Pilani, which is definitely the credit goes to the faculty, students and staff here. And today we have about 7,000 students on campus and about 300 plus faculty on roll. And this is a campus with also a lot of flora and fauna. And very recently we identified that we have about 250 species of birds, both migratory and those which are found here in Hyderabad campus. And we have started, making the database of this particular species of birds. And this is also something which is strength of this campus. And with this, I wish the organizing team for making this 4-day event to become a success. Thank you.
Thank you, ma'am. May I now request Professor Sonak Roy to kindly say a few words.
Guest of honor, co-founder and executive chairman of Bharat Biotech Padma Bhushan, Dr. Krishna Ella, director Professor Soumya Mukherjee, the dignitaries on stage and off stage, the key person for the conference, Professor Swati Biswas, and Professor Balram Ghosh, my dear colleagues and dear PhD students. A very, very warm greetings to all of you. It's truly a pleasure to be here in the inauguration of the Innovation in Translation Therapy and Targeted Drug Delivery Conference cum workshop. I understand that this is supported by SPARC, an initiative of Ministry of Education, Government of India, aiming to strengthen the research ecosystem and collaborations within the globally ranked international institutions. So I represent the Research and Innovation Office of BITS Pilani University, so I will just very quickly portray a snapshot of the activities, of the research activities, what goes across the campuses in the university. So since its inception as an excellent teaching institution, BITS Pilani always has worked a very, very unique path. Our strong industry linkages, whether through the practice school program or through the work integrated learning program, have always been our hallmark in this journey. More recently, with the growing focus on the research-led pedagogy, BITS has gained the recognition as a leading research-driven university in the country. And let me also highlight the fact that the NIRF 2024 ranking, the Department of Pharmacy was ranked 3rd. The 25 ranking is yet to come, and I do not see any suspicion that why you should not go and achieve the top ranking. So we foster a significant cross-disciplinary research initiative through the scheme we call that CDRF. We actively encourage our student-driven entrepreneurial program, which we call the Sparkle and the Solve. A few quick statistics to put the things in the perspective. So BITS Pilani is a home for around 18,000 undergraduate students across all the campuses and 2,500 PhD students, among which 44% are female. We also support more than 35,000 working professionals who are motivated to upskill through our program. We currently house around 1,150 dedicated faculty members to mentor and guide our students. On the research side, we are currently executing more than 550 extramural funding projects worth over something 550 crore. In the last calendar year alone, we have published over 3,000 research papers with an average citation of 20, and 25% of them are involved in the international collaborations. Not only in the research publications, last year we have filed 178 patents, and I can proudly say that 18 technologies have already been transferred to the industry, ranging from the electronic devices to the biotechnology. But having said this, when I look at the numbers very closely, it is only the 10% of the collaborations we have along the non-government industries, both in terms of research investigation as well as the capacity building. I believe this is the pan-India issue of the huge gap, the wide gap between the industry and the academia collaboration. And BITS Pilani, we always stand to bridge this gap. We welcome eagerly the partnership with industry, as I said, both in the collaboration research partnership as well as the capacity building. And when I say capacity building, let me highlight this fact that we run a very unique program called IMPACT, industry-mentored PhD in advanced and cutting-edge research. I believe that this particular platform, like this particular conference where academia, industry, the students come together for the technical discussions, can be the ideal ground to nurture the collaborations. And I wish a very, very stimulating and exciting technical sessions coming forward for the next 2 days and the workshop for next 2 days, and a very memorable experience to all of you who have come over here for the technical discussions as well as for the hospitality. Thank you so much.
Thank you, sir. I would now request Dr. Mohan Rao, Head of the Department of Biological Sciences, to kindly address the audience.
Friends, I am indeed pleased to be associated with this inaugural session, although rather unexpected. This is an excellent program of the Government of India, where international institutions and the Indian institutions work together, link, establish collaborations, and proceed in advanced research. This also gives us an opportunity to showcase our research and also benchmark with the international standards, which is rather essential in this current interdisciplinary research activities. My most important reason to be here today is Dr. Krishna. We know each other for a pretty long time. He is a vaccine guru of our country, and before COVID vaccine, they have produced a large number of vaccines for the world. I still remember when COVID was very rampant. I received a call from Krishna asking me whether we can get the viral samples so that they can start generating antibodies for the vaccine. At the time, I asked him to get in touch with the then director, CCMB, and for some reason that line did not work out. But eventually, National Institute of Virology, Pune, provided the sample, and the rest is history. All of you know what really happened. I again still distinctly remember the negativism in the country about the Indian vaccine, including the directors of national institutions and the presidents of the national academies were skeptic about the possibility of India making a vaccine, although many of us were very confident. I remember when I was on TV and explaining them that the vaccine would come within certain time. Soon after that, maybe 15-20 minutes after that, there was another program on TV in which one of the national scientists said that there's no way vaccine is going to come. So you have to be prepared to rather suffer the disease or die or keep the distances. That was the negativism. Even when vaccine was ready, there was a lot of negative thoughts about it. But I'm very happy and that Dr. Krishna showed rock solid confidence, and our country has really succeeded. And now we are global leaders. World looks at us as leaders in producing vaccines, and we provided solutions to many people. I only wish Dr. Krishna had a larger production facility. Whatever they could produce, maximum they could produce, they could supply to a large number of countries, including India. But if they had even bigger facility, it would have been even better. And I'm sure the country can be confident that any such eventuality, we have Krishna to help us. I'm very pleased that he has come here to talk to you. This is the best opportunity to understand and listen to the leaders in science so that we will be able to also lead. And some of the youngest people who are sitting here should also emulate such stalwarts in science. And I wish this conference a very good luck. And also the younger people to stimulate, inspire, get inspiration from the faculty who are doing their work here, and try to do the research which makes difference to the country. That's what is important. Country is looking for you. Thank you. All the best.
Thank you, sir, for your inspiring words. With this, we come to the conclusion of today's inaugural ceremony. On behalf of the organizing committee, I extend my heartfelt gratitude to all our esteemed dignitaries on stage for gracing the occasion and inspiring us with your words. We now request our respected dignitaries to kindly take the seats in the front row of the auditorium as we move forward with the next part of the program. Thank you.
We can start.
Yes, ma'am will introduce you. Ma'am will introduce you.
Introduction? Not required. I'm not a bridegroom now. Don't worry. Not required. Don't worry. Let us not waste time.
Sure.
Thank you. Thanks a lot. Thank you. Thank you. Really appreciate it. I think I'm the most senior, citizen here in this room. I'm 70 now, touching 70. I think even Dr. Mohan Rao will be younger than me. I think it's really honored to be here. I think Swati Biswas, she's got a Red Bull gene in her body, and she pushes the message in WhatsApp, and she knows that whether the message is delivered, and then when I see that, she knows that when I saw that also. So today is a world, different world. I think I saw her energy. Thanks to Director, Dean, Roy, and Vamsi, and Aarti, Yogeshwari, Dean. Thank you very much for being here and inviting me. I think I'll make it very simple, okay? All of you are very serious. I don't know why. I think, India, you have to, even with all the problem, you should keep smiling. You cry. You have to cry inside the heart, but keep smiling. That makes at least energetic level very high, okay? So don't get negative thinking, okay? Just for this thing. A little resounding. I'll try to see that a little bit. So I think I'm going to talk very few things, simple thing. And today the world is changing, totally changing. I'm seeing the technological angle and geopolitics, everything is changing the world. Today from brain intelligence to artificial intelligence. And you see oil world, which is oil was dividing the world. Now oil has become now electrical vehicles and hydrogen fuel. And also you see the coal has now replacing the solar and wind energy. And you look at iron. The iron was the important material. Now it is new rare earth materials have come into the picture. And also uranium getting replaced with thorium, which is coming from the sand. And lead, which is used in the battery industry, is now getting into solar and sodium batteries now. The whole science is changing. So you need to understand that. And I think in the last 2 years, the amazing thing happened in the post-COVID time and post-COVID time. So much changes happened in the technology. I'll tell you a few things. Human genomes can be sequenced in 5 hours. You give in the morning, you got an answer in the evening of the entire human genome. It used to take years before, now it has come to hours. So the whole science is changing. And the UK has come out, you can chemically, the entire chromosome of an organism can be synthesized chemically. You don't need a chromosome anymore. So I think the whole, the crystallization in the zero gravity, which is in space, how the structure it forms differently, and the molecule, whether small molecule or large molecule, how structure it forms in the zero gravity is another science. Elon Musk has come out, Neuralink, the way even a paralysis person can read and understand a lot of things. The whole science is changing. How is mixing the physics, artificial intelligence, robotic, every science integrating into chemical engineering, everything is integrating, neuroscience, everything integrating and come out in a different angle. And recently, PET, which we are drinking the water bottle here, this bottle can be converted, PET is converted into paracetamol with a two-step process by using a synthetic biology. Just simple synthetic biology. So you look at how the things is changing. So now I think innovation, people talk very, I don't think BITS Pilani can teach innovation. Neither IITs nor even MIT cannot teach innovation anywhere in the world. Nobody teaches. I think innovation is very simple. So here, how many Tamil kids are here? Any Tamil? Okay, Goyagashwari, good. So a few. I'll ask you, In Malayalam, how many people Malayalam? Malayalam, no, none. Okay, good. Malayalam, one lifting, slowly lifting. They're far away, they're slowly lifting. So I ask a simple question. Nallanna, you heard about Nallanna? Okay, you heard about it? Your grandmother said. Why it is called good oil? Do we know the science? We don't know. And in Telugu, it's called Manchinona. Okay, Nugul Nona means sesame oil is called Manchinona in Telugu. All South Indian language, in Kannada also, Vallanna. Okay, all 4 languages calls us good oil. My grandmother said, we are also saying, we are also scientists sitting in this room and talking about good oil. But why it is good oil? We don't understand. But the problem is, we take from US, we train and get into that perspective of lipid science and get into that mode. And that is not what innovation is. Innovation, you have to look at it here, what we have. And look at the other one, simple example, coconut oil. Everybody said coconut oil is very bad. You get a saturated fat, you get heart attack and all that. You look at Kerala people, they take a bath with the coconut oil, drink coconut oil, eat coconut oil. Every day is coconut oil. But they are healthy actually. They're very healthy. Suddenly some Netherlands has published a paper saying that it's good for Alzheimer's and Parkinson. Now, virgin coconut oil is exported to US now, from India and Thailand. Okay, entire virgin, everybody is now drinking 1 tablespoon of coconut oil now. Everybody is drinking. But same coconut is here, but we never understood the value of it. Simple things. I don't want to talk big science because my PhD boss in Wisconsin told me one thing. Think science in simplest language, then only you'll find the best solution in the world. If you think science in a complex manner, you will never find a solution. You'll never think that science can achieve anything. Think simple. That's important for some of the graduate students who are undergraduate here. Please understand. Think science simple. Don't complicate the life. And canola oil. You heard about canola oil? I know a lot of faculty will be coming from US and all that. Everybody knows canola? And do you know what is canola? Anybody knows what is canola? I know some of the many faculty would have used canola oil in US and all that. Okay, what is canola oil? It is nothing but mustard oil. Do you know that? Even ICR doesn't know that it's coming from canola is coming from mustard. 1962, Canada took from mustard seeds from India. They did a classical breeding, Brassica napus and Gensia, classical breeding to reduce the smell of that smell, ferric acid, whatever that smell, and brought it as they don't want to call mustard oil. If you call mustard oil, you compete with India. So they're called canola. Can means Canadian, wala means oil. Canola oil. See the branded? Is now 50% of agriculture export of Canada is only one canola oil. So where do we stand? We have mustard, North Indians, and particularly West Bengal, a lot of Bengali people here. And Bengali, without mustard oil, you don't eat anything. You don't eat anything. You look at all North Indian, East India pickles, all made with mustard oil, and South Indian pickle made with sesame oil. You see the contrast of culture. That culture has got science, but we are not realizing. We think doing that science is a low end. Actually, it's a very high end. This is the science we can compete with US. We can compete with Europe, but we don't realize the value of that. That's the sad part of it. And then we drink orange juice because Americans are drinking orange juice, Europeans are drinking orange juice, so we also should drink orange juice. You see Musambi juice, and you see Bale juice in UP, Bale, and black jamun, black jamun. Have you seen a juice anywhere in the country? You don't see that. Because Americans have not grown Musambi. If they had grown Musambi in sweet lime, they would have made that also, Musambi. So we have just changed our system. But look at it, in 1 month, the world is turned in the opposite direction. So United States of America has become United States of Asia now. Well, in 24 hours, all in less than 1 month. In 1 month, the world is geopolitics, changed from this direction to this direction. And the world is changing, very fast changing. So I think, what is going to happen in the future of innovation, I'm going to talk about entrepreneurship also, a lot of young kids. I'll share some of the stories of mine. And structural biology. I think the future is going to revolutionize the science. I'll give you some of the hypotheses, which doesn't have evidences, but I can only hypothesize. One of my course in Wisconsin is a scientific hypothesis. It's a course, one great course. It's one of the toughest course. how to think science, scientific methods, scientific hypothesis. Okay? So, structural biology. It would have taken 200 years, millions of scientists, postdocs, to solve all the life sciences proteins to structural biology to solve. Now, all are solved in less than 2 years. With AlphaFold 3 or even the University of Washington, Seattle software, whatever it is, they've changed the whole science. Now, you can do structurally, predict almost 95%, 98% accuracy by using structural biology with the software. So now what is going to happen now? What will happen? Now, you will find an enzyme that can digest this bottle in less than 1 hour. You'll find it. That's my hypothesis. So don't ask me evidences. So you always hypothesis, you design based on that hypothesis. People start visualizing how structural biology, thermostable, fast degrading, and selective to the PET, ethylene binding. All that you're going to come out somewhere. Biosimilars will be wiped out. Entire biosimilar monoclonal antibody, which are there in the globally right now, will be wiped out. You will suppose if this protein monoclonal antibody, this is the length of the protein. Now they'll come out based on structural biology, short sequence, only this much requirement. Only this much requirement. And if that region is enough to make a protein and a short peptide, which can bind to the receptor in a pharmacokinetics, very high binding capacity. So you basically, you bring the dosage down, protein concentration down, and you'll have more selective, less adverse reaction. Less adverse reaction. So you'll change the whole field for monoclonal antibody field itself, will change everything, science. That's my hypothesis. And it's going to change. And you know, people talk about quantum biology and all that. I don't understand much about the quantum biology. I'll tell you, Indians' forefathers, they discovered the zero. And that is a quantum biology, good example of quantum biology, quantum science, quantum mathematics, whatever you call. You know, after that, Egypt added the script to that next one. Greek added another script to that. Each one changed it differently after zero, added some other symbols next to that and all that. That is a different game. But Ramanujan, who is uneducated, he was not even a clerk, he was a clerk in the port trust, and he visualized the mathematics, even now you are not able to solve. So where do you need? So your brain his brain is really quantum mathematics, literally quantum math. We need to understand the value of that itself. About my entrepreneurship, it's very simple. I was in US, I'm from Tamil Nadu. I was in US. I said, my mother was always used to tell me, son, you have a 9-inch stomach. How much ever money you make, you can't eat more than the $1 also in a day. So you come back, do whatever I like to do. I'll fund you, I'll support you. I'm from agriculture family. I'll grow vegetables and fruits and feed you, not even starving a day. I think my mother was really forced me to come back to India. And I think that's a time when the reason I was never had a vaccine experience, zero knowledge of vaccine experience. The only knowledge I had, a domain knowledge, was a skill set, was yeast molecular biology. So I was in late 80s, I can precisely put the gene out and gene in. That was gene knockout. I was in 89, I was working on knockouts on the yeast. That one field helped me, okay, what can I do in yeast molecular biology? Then hepatitis B came into picture. Hepatitis B produced in yeast. And that is how I got into vaccine field. Otherwise, I have zero knowledge in vaccine. I'm basically an agriculture graduate. I didn't tell you that. I'm an agriculture graduate, and then went to US and changed my field in signal transduction, molecular biology, virology, and all that thing. That's a different matter. But what is important is that one skill set is what led to me this one. So I think the most important in the life, you need a skill set. Chinese have done extremely well on the skill set. They can't even speak English, but their skill set is amazing. That's something we need to learn as India. We are good in knowledge, but not in the skill set. That's very important. We made various vaccine, rotavirus vaccine. We made with Indo-US bilateral program. We export more than 121 countries. So I want to ask here, a lot of ladies here. I want to ask, when you get married, you want to give their newborn baby for a clinical trial. How many are willing to do it? I ask a simple question. Not even a single lady lifting the hand. So how do you build a science innovation in this country? You want Americans to do clinical trial in their American babies, and after the product gets here, then you want to give it to your babies. Is that fair? It's not fair. You want innovation to happen, clinical research is the most fundamental important. Whether any drug discovery, the most important fundamental is the clinical research. And we had a Gates Foundation funding for this project, and CDC Atlanta and all of them were involved, Stanford, everybody's involved. They said, we should not bribe the mothers. That's one of the conditions. So I had to recruit 25,000 pregnant women to give 10,000 babies. And then no bribing. You see, imagine. Because India never done a clinical trial, clinical research at all. They do clinical trial, okay, you inject ampicillin here, and you inject Pfizer ampicillin here, both are equal. That's a clinical trial in this country. Okay? It's a sort of bioequivalence or immunogenicity comparison. That's all the difference in the clinical. That is not the clinical research at all, to be frank with you. The clinical research is in the field situation. You need to have a double blind. You have placebo, some children's. We had 3 years, University of Pennsylvania, ethical committee issue, whether it should be given placebo at all. One of the mRNA vaccines in US, why it's getting controversy, there is no placebo in the clinical trial. So the question is asking now, without placebo, how do you predict the adverse reactions, what has happened in the trial? I think we are the only company in the world, Covaxin is the only one, we had a double blind and we had a placebo trial. Even US government appreciated that we had a placebo in our clinical trial at 1:3 ratio. And I think, what is important, we made a rota, and pregnant mothers, we gave a health care benefit to the mother and child for 3 years. And they've been given the best vaccination, all the vaccination to child. Whichever the Mukesh Ambani would have got, their children would have got, we give the same treatment to their mother and child. That is the incentive we given as insurance. We put all those things. That's the history. Thanks to Dr. Bond, who was really visionary, and he helped us, and Mohan Rao knows very well Dr. Bond. And he is really visionary, helped us to make it. Otherwise, we would not have made it. As a small startup company, would not have made it. So there's so much of bilateral program helped us on that. And typhoid, we are third company in the world. We made typhoid conjugate vaccine. The children who are most poor people in India are the one who licks the floor, who gets contaminated food. They get the typhoid. And the antibiotic abuse. If you go to a doctor, pediatrician, they give 3 antibiotics, unfortunately. Because they can't tolerate the mother and grandmother's torture. So they want to get 3 antibiotics, so the mother and grandmother will be shut down. So I think it's so much of abuse happening in the country, and we said we make a typhoid conjugate. First time in the world, we made it. And then, now I want to ask gentlemen here. Now, you have a double-blind studies. Now, how many people are willing to take a oral typhoid, virulent bacteria, typhoid, drinking? How many? Oh, Vamsi is ready. One person. I think he's only one. And lady is also lifting. Good. So I think she found that my baby is okay, but I can take it. I don't want my baby, but I'll take it. I'm just joking, don't worry, okay? So I think good. So I think, we can't do it in a trial in the human challenge in India. We have to go to Oxford. We go to Oxford, 120 people. We did a double-blind human challenge studies. We got 87% efficacy. We published in Lancet, New England Journal of Medicine, all that history. Now then, when you develop India innovation, then a lot of barrier comes in at a global level. The next global, you're done in, in the laboratory condition, in the human challenge studies. Will it work in the field situation? You give it a vaccination. So we had to do 3 million children, thanks to Gates Foundation. They funded us $36 million. Even the human challenge studies was funded by Gates Foundation. So we did in 9 countries, we did the effectiveness, 3 million children, vaccinated, non-vaccinated children, and then followed up for 5 years whether they get typhoid. So we found 85 to 92% effectiveness. It's called effectiveness, not the efficacy. It's the effectiveness studies. Why I'm saying this one, please understand, Indian R&D is not anymore in the primitive. That's my perspective. It is not to brag you. I'm not a listed company that to buy a shares here. So I'm telling you this one, understand the complexity of the clinical research. And without that, you can't do drug discovery to move to next level. Never. Okay? I think that's a major bottleneck in this country. And typhoid conjugate, we did in 3 million children. We proved, now we export a lot of African countries and all that. And chikungunya, we were the first one to sequence the genome in India, not even NIV Pune. We did the first one. And then we predicted Zika might come to India. The chikungunya was, we predicted chikungunya based on sequence analysis. It indicated it came from a French island, Madagascar to India. Then we asked next question, okay, if it came from Madagascar to India, through mosquitoes, what will come next from Madagascar? Because there was only 4 diseases: chikungunya, dengue, yellow fever. Unknown disease was Zika. 1942, they published a paper that there was a Zika spelling. Nobody knows. CDC was not even aware of Zika. We were the first one to make a Zika virus strain. Okay, and then made a vaccine in 2014, and 15 July, we filed a patent. And December 2015, Brazil incidence epidemic came. And it is getting into problems slowly in India also now, pregnant lady. Any pregnant lady gets it, it's a more dangerous, whether it's intellectual IQ level or the neuropathic problem in the Zika problem. So we are working now, and that got killed by, thanks to Government of India regulatory system, choked us, both the project. If it is the Americans made it, they would have made it. I'm telling you, if I made this vaccine in Singapore, I would have got at least $100 million from Singapore government. Honestly, I would have got $100 million for projects. But we pulled out regulatory system, always suspicious of regulation. Chikungunya, they made us emergency phase 1 trial in KME Pune. KME in Bombay. So that means it took phase 1, it took 3.5 years. That means the fellow who is accident injured or heart attack patient, along with a healthy fellow, will be sitting next to it for vaccination. That's how they approved us the trial permission. You see, some of the people you are in the committee, please be aware of it, the global science. You need to help the countries to move forward. Don't sit in the committee, be like I have not done how can that bugger can do the science? Don't look like that. You be proactive, help them. Make the country to be progress. Society should progress in innovation. That's important. Every member I told, University of Hyderabad, some of them sit in RCGM and all the lousy committees, okay? They don't do progressively. And they're choking the science of this country. And I think, it's good for me, those committees, because I've reached a level that I don't need any competition. I don't want any startups to survive. In that way, if you look at it, yes, it's good. But it is not what we want as a country, as a society we wanted. I think we need to change that. And I think Covaxin, we did only one thing. I was believing that inactivated vaccine, when you give it we are giving the vaccine to the healthy people, not infected people. When you give it to healthy people, the most 3 fundamentals are important in a human, when you give it to healthy people. Safety, safety, safety. In the COVID time, everybody was talking about the efficacy. Oh, mRNA vaccine got 100% efficacy. They designed intelligently the clinical trial. At that time, when Wuhan time, without placebo, you did, and it showed 100% efficacy. You intelligently designed the clinical trial, you show 100% efficacy. Now, and we spent about 700 crores on the clinical trial alone. The trial cost 700 crores, including Equia, the multinational company. We hired them site management. Okay, 700 crores. I never took 1 rupee also from the government of India. I said, as a scientist, I need to contribute back to the country. And I don't want to exploit the government of India, or I don't want to exploit the society at that time. We spent 700 crores, we took a risk, we said, let's fail, doesn't matter. But I want Indian science to succeed. That's the only mission we did. And we now 27 publications on Covaxin alone. In that 9 Lancet, 2 Nature publications. Remaining all top journals. Okay? Nothing less than science. See, now you see how geopolitics hurt me that time, the COVID time. And the scientist's enemy in this country is scientist only, not politician. Scientist's enemy is only scientist. Each scientist jokers gets into the TV channel. And, soft target is Indian company. No, you can't talk Pfizer, you can't talk Moderna, you can't talk AstraZeneca. Okay? The soft target is Indian company. Oh, Krishna has not published a paper yet. I mean, as if my grandfather, Lancet journal is my grandfather's property, I can publish in 1 day. The stupidity of these people. And every scientist gets in the TV channel, talk against Indian company and Indian vaccine. You know what it says? The nice story is, exactly in 2 months back, US government has announced mRNA vaccine is not good. Now $500 million allocated for inactivated vaccine. Is it enough now? Look at the internet, science article. There's a science article, July article. Look at it. Inactivated vaccine, they're now funding $500 million. They strategically said, very strategically, they said people lost trust in mRNA vaccine, so therefore we want to fund the inactivated vaccine. So you rephrase the statement that some vaccines causing adverse reaction, that's why we're not funding. That gets into legal disputes. It's a strategic game. But we did a phase 1-2 in US on a Covaxin. And then US FDA said, we are not going to approve your vaccine because we can't create a competition with mRNA with you. So please, after 2, please stop it, don't continue. So that was fair enough for us. At least we stopped after phase 2, we stopped the US phase 2 trials. I think, I look at one thing, I'm telling honestly, I started the business based on not on the money. I look at what society problem. I look at Nagpur farmer commit suicide. We have too many economists telling sustainable agriculture, that agriculture, this agriculture. Farmer is an entrepreneur. He puts 100 rupees, you need to get 150 rupees. Do you need any big scientist, macroeconomics to tell in this country? Nothing. What he produces, you should get the price. He's an entrepreneur. And today, Nagpur farmer commits suicide because one single crop, cotton, if the monsoon fails, the cotton crop fails, and they commit suicide. You come to same rainfall area, Kolar district, come to Karnataka, they don't commit suicide. We ask simple question, why is the Kolar farmer doesn't commit suicide? They depend on allied agriculture, dairy, poultry, sericulture. Because of the poor other side, even crop fails, they sustain because of allied agriculture. I want to see the faculty, don't look at human alone, human research alone. There are a lot of other fields opens up for you, whether pharmacology, molecular biology, please get out of this human. Human is nothing now. Human, you have to get out of the human, get into the other side of the science itself. And we see that, okay, we should do something innovation. Who is there? Public sector, government company. The government companies cannot innovate in this country. They're only busy solving today's problem. So we set up a biovet in Bangalore on solving some of the problems. We are the first one to come out with the lumpy skin vaccine for the cattle, which are reduced 30% of the milk production down. We are the largest containment facility in the world. We are BSL-4 containment production facility in Bangalore, which is underground. It's one of the best engineering. Some of your faculty should go see the Bangalore facility and also 2 BSL-3 facilities in Bharatbhate. We are 10 minutes away from your BITS Pilani. But your BITS Pilani faculty are all busy with your own routine problem, writing a report to ICMR, DBT, DST, only reports. No science moves. I think you need to be out of the mind to think. So my message is to faculty. And I was in ISA Trivandrum, chief guest for the convocation. So the director Murthy asked me to talk to the faculty. So you tell me now, Yogeshwar is a dean, and Anita Dar is become a professor now. Tell me, after professor, how many director posts are there in the country? There are 5, 6. Nothing else to do it. This is the time the professor, because I'm telling this one, because assistant professor said, if I don't publish a paper, I don't get associate professor, I don't get a professor post. Fine, understood. Then I told him, okay, you're all 36, 37 years old, you're becoming a professor. What else for you in the life? You can't get Dr. Mukherjee's post. How many people will be competing with the Mukherjee post? So very few director posts. There's nothing for you to lose life. Get on to some risk taking. Do something. At least professor can take a risk. And that will be a motivation for a junior faculty. That's a message I want to give it to the faculty. And I think the country offers so much opportunity. But don't bring your US concept, wherever you're trained in Europe or US, don't bring that concept to India. Please start looking at micro details, what the society problems are. Take that problem. In US, most of the research, I was in the Governor Thompson's Senate meeting in the Madison, Wisconsin, where the funding based on the society problem, not based on the faculty's interest. Okay? So I will tell that is a different story. So then I said, the most of the biotechnology sector concentrated only south and west, nothing in east. And all Bengali people, they're all coming to south. Where the nothing Mamata Banerjee destroyed entire Calcutta. All east is gone. So I said we should move something to east. I don't want to be just south-centric person. So we invested 1,500 crores in Bhubaneswar, in Orissa, and we created one of the most modern plant. I think some of the pharmacology people should visit the most digitalized facility. And then, 3 months back, Singapore President, Singapore Transport Minister, 8 members of Parliament have visited my Bhubaneswar plant, Jamie. Visited the Bhubaneswar plant. They want to see how India creates products. They want to understand how India creates a product. And then how you create innovation. How entrepreneurship is driving this country. This country is driven by entrepreneurs, not by government. China is driven by government. China drives the entrepreneurs there. But India is just opposite. Entrepreneurs drives it. So today, maybe I can talk against the government. It's not, government is part of my system. Why I'm saying, I don't want young generation to leave and go to US. We need to create the ecosystem for this country to ourselves. Everyone in this room are responsible to create the ecosystem. Don't expect the Prime Minister Modi to create an ecosystem. Everyone, one of you sitting in the committee, you can also create that ecosystem. You have to be bold enough, magnanimous enough to understand that. And I think I'll leave it there. And thank you very much for giving me opportunity. And I think India is going to be tremendously going to opportunity with the new geopolitics that is happening. That opens up the game so much to all of us. Good luck to you. Thank you very much for the opportunity.
Thank you, sir. What a truly inspiring talk, and I think the 20 minutes just passed by like a second or a minute. So I was so, personally I'm so impressed, this is maybe a lifetime hearing something that will truly inspire me, and of course all of you who are here, I think very inspiring. Something that really, something that you said about the canola oil. Really, for 10 years in the USA, we were all using the canola oil and never thought that this is coming from mustard oil. So we were using it, but never knowing it actually that's it. And of course, the way that you said that we have to actually think about the problems in India, and we have just not a stereotypical thing like what we have learned from USA, and we should carry forward that. I think we have to actually really change that. So this is a really life-changing thing for me also.
I just want to say, all faculty, when you publish all the 200 journal, 300 journal, once you retire, your own children will not remember the publication. That's the sad part of it, okay? So please do something for the country, for society. You'll achieve bigger.
Thank you so much, sir.
Gift, I'm waiting for gift.
Yes. May I request Professor Mukherjee, Professor Yogeshwari?
I'll go and receive there itself. You don't have to walk. I'll come there. No, come on.
I said there is a...
Cameraman, come on, take my photo. My wife doesn't see my photo properly. Talk loud. Talk a little loud. Sure. You need a Red Bull drink?
Now it's time to start our scientific session.
We would like to invite our first plenary speaker, Professor Jimmy Shia from NTU Singapore. Professor Shia is the President's Chair Professor in the School of Mechanical and Aerospace Engineering and the School of Chemistry, Chemical Engineering and Biotechnology at Nanyang Institute Technological University, NTU Singapore. His research interests include mechanics of soft materials, mechanics of plant organ morphogenesis and morphing metamaterials, mechanics of mammalian cell and micro and nanotechnologies in mechanical devices, and structures, smart adhesive systems, and soft robotics. I now request Professor Jimmy to come on the stage and share his views on biomarker-triggered hydrogel swelling for ultrasensitive biosensing.
as they're setting up the computer, let me just say a few words. I've known Dr. Ella for 10 years now.
Which is a folder.
It's always been very inspiring to hear the thing I like most about his speech is the positive attitude, the can-do attitude. Doesn't show up over there for some reason. Good. Okay. So thank you, Dr. Ella, for this inspiring speech, and it's a great pleasure to have known you. You know, we met each other for the first time in 2015. I remember we had dinner together, and ever since then, it's always great to see you again. Okay, so I'm going to tell a relatively easy and short story. The whole thing started with collaboration with some people in China at Zhejiang University, especially Professor Ping Wang and Professor Qingjun Liu. And we've published, or they have collectively, we've published a couple of books and also a few review articles. The main topic we're interested in is this mechano biosensor. So if you ask what is mechano biosensor, first of all, chemical reaction is involved and mechanical deformation is involved. And in order to become a sensor, we have to have quantitative detection. Okay? So the whole thing together makes a biosensor or mechano biosensor. Okay? Let me say a few words about what are these chemical reactions that we're talking about, or mechanical deformation detection. Chemical reaction, we're referring to biomarker sensitive hydrogels in this case. And mechanical deformation is a deformation that due to sensing of these biomarkers to make mechanical change. In this case, it's a swelling of the hydrogel. And in the case of detection, we use ultrasound or ultrasound resonance frequency shift for that detection. Okay. And altogether, I'm hoping that by the end of this lecture, I'll show you that we do have an ultra-sensitive biosensor. Okay, so if you have something that relies on, for example, environmental and physical stimuli, generally speaking, the signals are strong, or stimulation intensity is strong, and the hydrogel deformation is, you can stretch it, it can be large, 100% and so on, and the response is spontaneous. However, if you want to use biochemical stimulation, generally speaking, the intensity of the stimulation is subtle, and the deformation is minor, and the response is sometimes slow. Okay, so how do you overcome these difficulties when you try to detect some biomarkers? What you need to do is, first of all, you have to have biomarker-sensitive polymer network, and in our case, you can design a metamaterial that is sensitive to very small strain. And also, you want to develop the device that would have very fast response. Okay, so these are the requirements. In the case of chemical reaction, in this case, we actually make use of a particular principle, which is competitive binding. Okay, what is competitive binding? Okay, so we have a two-phase polymer. In this polymeric material, a hydrogel, we have the carbon-carbon double bonds, and we also have the secondary binding, which is the receptor ligand, what we call immobilized binding. Okay, for example, in this case, we have antigen-antibody binding that would serve as a secondary binding. Okay, so when the hydrogel is formed, you have cross-links, the primary binding, and you have the secondary binding. However, if when you put in a biomarker, that biomarker has a stronger affinity to the antibody, what would happen is it would release the secondary binding, and as a result of this, the hydrogel swells. Okay, that's the deformation or mechanical deformation part. The same principle can be used for other types of competitive binding. For example, you can have nucleic acid. This is specialty of Dr. Ella. And you have secondary binding to the microRNA. But if you have a biomarker that has a stronger affinity to the microRNA, and you release these binding, and as a result of this, it expands or swells. Okay. Similarly, with glucose, same idea. Okay. In the case of glucose, it's also even more interesting. Okay. You have some binding sites for the glucose, but these bindings, the glucose would bind to these. However, if you have a low concentration of glucose, it would bind multiple sites. As a result of that, the hydrogel shrinks. But if you have a high concentration of glucose, it expands. So you have all these so-called competitive binding process that would give you different mechanical deformation. Okay. How do you use that mechanical deformation? You use that to detect these biomarkers. Okay. And in this case, I gave you one example where using this CD63 as a particular biomarker, and you can see that with the CD63 in there, the swelling strain increases as a function of time. And in this case, it's hundreds of seconds or several minutes. Okay? And what is interesting or benefiting is this swelling strain is also a function of concentration of this biomarker. So you can use this, and if you can measure the mechanical deformation, you can use this as a sensor to sense certain biomarkers. However, there are two challenges in this case. One, how do you measure a mechanical deformation or the swelling? Okay, this is one challenge. Second challenge is, generally speaking, if you have a block or a big chunk of hydrogel, the response is very slow. Okay. So what we did was, to solve these problems, we developed the metamaterial hydrogel. Okay. The purpose of doing that is to use nonlinear behavior to amplify the effect. What nonlinear behavior we're interested in this case, we're interested in buckling. Okay. Buckling is intrinsically a nonlinear behavior which transforms a symmetric type of situation to an asymmetric type of situation under deformation. This was done by a colleague of mine, Katia Bertoldi at Harvard University. They've shown that using this buckling approach, one can transfer these metamaterials and to obtain, first of all, very large deformation, and secondly, they can even have something like negative Poisson's ratio, which is a mechanical property of these materials. So this is something that we make use of, this nonlinear or local buckling pattern formation to amplify the small deformation, to amplify the signal. Okay. One other effect of this is once you have this nonlinear deformation, then the acoustic response of the material will change. For example, in this case, before buckling occurs, before you apply a deformation, you don't have any what we call bend gaps. Okay? But upon deformation, when it buckles to an interesting shape, then you form band gaps. Band gaps are the frequencies where the transmittance is very low or waves cannot pass through those frequencies. Okay? And in fact, this ultrasonic detection method has been used by many people. Let me show you. These are fairly recent paper, 2024 Nature paper, 2025 Nature Biomedical Engineering paper. They all use this band gap concept or the acoustic hydrogel to detect certain types of mechanical deformation, in this case, swelling. Okay? So these are the basic concepts or basic tools that we use to achieve one technology we called M-basis, or metamaterial buckling assistant surface acoustic wave enabled sensor. We call it M-basis. Okay. All right. So what is M-basis? First, we can design these hydrogels to achieve these competitive binding. Okay. And this is what I've already shown, the basic concept, but you can also see that for certain types of, for example, microRNA, it's very specific. For this microRNA 222, it's very sensitive. With mutation 1, it's less sensitive. Mutation 5, it's not very sensitive at all. And it's roughly about the same level as DI water. Okay? So it's very specific to this particular microRNA. Or in the case of glucose, you can show that as a function of concentration, glucose concentration, you can have first shrinkage of these hydrogels, then expansion or swelling. And in fact, this can be tested repeatedly. So if you put it back in DI water, it goes back. And if you put it in 10 millimole of glucose, and it has a tremendous shrinkage or strain. Okay. So this is the basic concept of competitive binding. And the other one we looked into is making use of this metamaterial, which is a patent material. And this is a finite element simulation result that showed that if you swell this material but constrain on the border, it goes up, the stress goes up, but at a certain point, suddenly it buckles. Okay? The uniform structure becomes interesting structure. And as it buckles, the stress goes down suddenly. Okay? And you can also have, in our case, three-dimensional, or you have a thin sheet of this material, hydrogel material, as a substrate, and you have this matter material on top, and the buckling strain can be manipulated. In this case, it's about 11%. You reach buckling strain. But with the 3D, it becomes very sensitive. Once it buckles, the stress changes tremendously. Okay? So these are all simulation results. And now the question is, how do you implement in the real device? So the matter material, let me make the statement here, plays two roles. One, whatever deformation, swelling you have, because of this buckling effect, you amplify that effect. Okay? Even small amount of swelling, 10% or so, and because of the buckling effect, it's amplified. Okay? So you get big signal. The second thing is, this is a structure like walls, okay, thin walls. And the entire structure of this has a dimension of roughly about millimeter or so, okay? So each one of these walls has a dimension of tens of microns. Because of that, the time response becomes very fast. So you put the biomarker in, or the solution in, the diffusion is very fast because it's smaller dimension, and it can detect these biomarkers within a very short period of time. Okay, and I will not repeat this, but one way we can measure it is to look at the rotation angle over here. So when it buckles, it rotates, and we can measure the rotation angle and to calibrate that as a function of strain. Okay. Another challenge we faced during the development of the sensor was, we know for a good sensor, you want it to be very sensitive. So I'm measuring rotational angle as a function of swelling strain. And you want to use a region where the rotation angle is very sensitive to the swelling strain. Okay? So this is a good sensor because it's sensitive. However, if you increase, in this case, if you want to really reach the biomarker strain, the strain is large, swelling strain is large. So you reach that region over here, so the rotation angle would not be very sensitive to the swelling strain. So in this case, it's not a very good sensor. So the student or the postdoc actually came up with the idea that we want to use the sensitive part of this, but for larger strain. So what he did was he left the gap, or the constraint would occur later, not immediately. So he left the gap over there, so the initial swelling does not cause it to buckle until you reach certain strain. Okay? And in that case, you can fully use of this sensitive part of this biosensor. Actually, here's one demonstration of this. Without using the sensitive, without the gap, you can sense for this particular biomarker concentration of maybe 10 to the first power, 10 to the second power picogram per milliliter. But with the gap, you can expand that sensitive region to a much wider region. Okay, so these are all details. But another thing we did was to use this acoustic or band gap to as a as a quantitative measure to measure the change of strain. So when it buckles, the band gap changes, and we can measure, for example, the shift of the frequency to measure whether it buckles or not. In other words, to measure whether it senses certain deformation, mechanical deformation or not. In this case, we can see that if you have a particular surface acoustic wave at this particular frequency for the metamaterial, the transmittance is very good, but when it buckles, nothing gone through, okay? And that can be characterized by the frequency shift. Okay, so all these things can be done. Let me show you the difference between just a film and a metamaterial. For a regular film, you can put, for example, PBS, a different concentration of this biomarker into it and measure the frequency shift. For regular film, the frequency shift is very minor. With the metamaterial, it's significantly amplified. Okay, at each of these points, you can measure the frequency shift easily. As a result, you can sense the biomarker with a much higher sensitivity. Okay. Okay. Here's the calibration of this for CD63. You have the sensitivity like this. For microRNA, 222, and for glucose, and so on. Okay. And in fact, here I show you the saturated frequency shift for these three different biomarkers, CD63, glucose, and microRNA 222. I also show you the how specific this particular sensor could be. Okay, this is all by design. For example, this microRNA 222, you have a huge shift of frequency, so you can sense it. But microRNA 221, 141, 21, no response, very specific. Okay. Similarly, for glucose, lactone, there are very different responses. So these can be designed to have very specific sensitive measurement. Okay. Another thing I can show you is the sensitivity and the detection speed. Okay. The axis over here is if you move from right to left, it's longer time. Also, if you move from top to down, it becomes higher and higher concentration or limit of detection. Okay, so top is sensitive, and to the right is shorter time. So you can see that for all these other biomarkers, or for the same biomarker, for all these other biosensor technologies, ours is both more sensitive and faster response. Okay. Okay. So I don't have to repeat this. This is the device that we made. Okay. It's a point-of-care device. It has a dimension of something like 5 centimeter by 4 centimeter by 3. So you can hold it in your palm. Okay, and you have the sensor over here. You have the inlet-outlet microfluidic channel over there, and essentially you can use this, and you can have acoustic waves. You can measure the biomarker. Okay, so good. So we can use this. We can publish a paper. Let me go back to Dr. Eller's point. What is this used for? Can you use it to detect something? let me show you what we can do. So we tried. This is, we're trying to use this to detect HSV-2 virus. HSV-2 virus is a herpes virus. It's a sexually transmitted disease. So patients very often are very reluctant to go to the doctors. Okay? So we have a collection of roughly about 34 patients. Okay? This HSV-2 virus is the most dangerous virus. And there's a HSV-1 virus, which is much less dangerous. What is also important for this particular virus is it's a recurring disease. It's a chronic disease. In other words, you cannot cure it. It flares up from time to time. Okay? So what we thought was, we have a device. We can just give it to the patient. Okay? They don't have to come to the hospital for treatment unless they detect that they're positive. The disease flares up. Okay? And so we use our sensor to detect this. Okay? With all 34 patients, first of all, this particular, the blue curve over here is the gold standard that hospitals use. It's called ELISA. Okay? And our sensor is clearly much more sensitive. Okay? With very low concentration, we can sense it, but also covers a wide range. We can also tell whether the concentration of the virus load is high or not. Okay? And several things we did. First, we have a control of healthy people, no signal. We have a few patients, number 4, 5, and number 33, and so on, with HSV-1, very little signal. But with HSV-2 positive patients, known positive patients, and we have very strong signal. So good, this sensor is useful. And we can also, for this particular case, have a good indication of how serious the patient is being infected. So it's actually, you can tell the concentration of this particular virus, and by measuring the frequency shift, you can tell how serious the disease is. So doctors can treat the patient with the proper treatment. Okay? Another thing we did was this thing. Okay? We have the regular patient. We know, the virus over there. We dilute it 10 times. No problem. We can still detect that. We dilute it 100 times. It's still okay. If you dilute it 1,000 times and above, there's no effect. So by this particular technique, or using our technology, not only we can detect it fast, but we can also, if you want to say that, you have many patients. I think during the COVID, it's a major problem, right? You have so many people all doing the tests, and you don't want to do every single one of them, but you group them into 10, into 50, and so on. And if it's positive, then you identify one of the 50 or 2 of the 50 are affected, okay, or infected. So this is one thing that we thought would be very useful. And this is other thing that measure the viral load, and using the standard technology, ELISA, comparing to our frequency shift of our M-base device, okay? And they compare very well. Okay, so and another thing we did was to compare our device with the DNA-based or measuring technique or real-time PCR and so on, and they also compare very well. Okay, so with our device, I think we have a fast response time, we have a high sensitivity, we have very good specificity. Okay, so let me conclude by saying that we have developed, a device or we use technology to design this biomarker-sensitive hydrogel metamaterial and use surface acoustic waves as a detection method to measure the biomarker. And we have been able to have a fast response, a very high sensitivity, and the detection method is validated for one particular disease, which is HSC2. Okay. And with that, let me acknowledge the student or the postdoc who did the work, Xin Li, and Xiaofei Zhu, a postdoc. She did the finite element simulation, and the acoustic wave calculation was done by Dr. Wenxin Zhou, and also the collaborator on this project is Professor Jingjun Liu at Zhejiang University. Let me acknowledge the support by Nanyang Technological University and Zhejiang University on this. We're also supported by some hospital. Okay. With that, I thank you for your attention. I'd be happy to take some questions. Thank you very much.
Thank you, Professor Shia, for setting the perfect tone for our symposium. It's our immense pleasure that you are here for the second time in our campus. Also, let's have a quick question and answer section.
So this is an excellent technique. One curiosity that you will have in a biological sample many other molecules present here.
Oh, okay,
yeah. Okay. So in a biological sample, you will have many other molecules present other than your analytes what you would like to detect. And hydrogels are many times sensitive to many other ions and things like that. So
good question. Very good question. So
how you can sort of be specific in that case?
Let me go back to one of the slides that I have over here. So it all depends on the design. Okay. So I mentioned that this is based on the principle of competitive binding. So you can design your hydrogel in such a way that as long as you can have competitive binding between these two different materials, let's call it one is antibody antigen, the other one is ligand receptor. So as long as you can design your material to have the right competitive binding, no problem. Okay, so it's not restricted to the biomarkers that we looked into. Okay, so we may not be able to find the right pairs to do all these things, but it's up to the chemist or biochemist to find the right binding. Okay, so to answer your question, certainly the principle is universal. The technology can be applied to many different molecules as long as you can find the right binding, right competition. Thank you for the question.
One question from my side.
Yeah.
So I noted that some of your positive controls did not have a signal. Is that because of the absence of biomarkers in those particular samples, or was it that certain variants in the biomarker may not be picked up if the antibody that you're using is not sensitive to that particular variant?
The last part of the question, maybe?
So the recognition of the biosensor by your system, if there is a variant that is there due to some mutation, would it still be picked up by the sensor?
You're asking about the release of these bindings?
Yes.
Okay. Okay, so maybe let me go to one of the earlier slides. This may be a better one. Okay, so in the case of certain biomarker, and the concentration of these biomarkers would, let's say you have a very low concentration, then you release some of these secondary binding. So that gives rise to smaller swelling. Okay, so the sensitivity of that comes in from what is the concentration of biomarker, how many of these bonds can you release. Okay, I don't know whether that answered your question.
Yeah, okay. I think I heard also the following his question. What we see is some of the healthy individuals where you should not be picking up, you seem to be having some false negatives. Are there any false negatives? That's one question. And second question is, you're using the gels. These are gel is a POC, point of care device. But what about acoustic system that needs to be detected? You need an acoustic system to do that. So you have two components. One is your gel. This gel will be POC, point of care unit, which you can take to patient. But detection, you need an acoustic system. So where is the acoustic system?
Okay, so good question. I think you're referring to these. Some patients, they don't have much signal, right? Okay, so these are positive or false negatives. Okay. Let me take it back and saying that this is preliminary. Okay, we only have tested, for example, 34 patients. Okay, and we need to go back and look into these, maybe perhaps using machine learning, big data, and to identify what exactly happened over there. Okay, right now it's a preliminary results. We're not claiming that it's it's mature. It's still preliminary. So we haven't looked into those cases carefully enough yet. But we will do that to make sure that we don't really miss anybody who are seriously ill. It could be the case that these cases are, or the design of the hydrogel are in such a way that it's not that sensitive to certain types of patients. It could also be the case that the viral load for these patients are relatively low. So we don't know which one is the case. But one thing we can be sure about is we don't have false positive. Okay, so these are healthy people. We don't see any signal. And for most of the patients, vast majority of the patients, we see strong signal. Okay. Second part of the question.
System, what we have to measure the acoustic wave. There must be a machine to do that. Is that machine part of the package?
Device itself?
Yes.
This one?
So where do we measure the acoustic signal?
Oh. Okay. So we're measuring the acoustic signal. There are connectors or electrodes somewhere over here.
No, my point is, this is POC. If this is POC, you need to have a device along with it.
Right.
So would it be easy?
You have to have another
system.
Yeah, another system to go with it, which is signal generator and detection.
Yeah.
Okay. That's actually another very good question. So for this thing, we need to have some equipment in order to do this. This is actually one major obstacles that we want to overcome eventually. So how do you do that? Maybe the reason we came up with the acoustic devices, recently many people are interested in that, and we know that it's very sensitive for this buckling nonlinear deformation behavior. But, we can have other types of detection as long as they're quantitative. Okay. Acoustic device, you do need some function generator, you do need a detector. Very good question.
Sorry for interrupting. We are running out of time. Thank you so much, Professor Shia, for being here. I request Dr. Mohan Rao to felicitate Dr. Shia.
Thank you, sir. I would now request all to join us for a group photo outside the auditorium. We will now pause for a coffee break after that, and please rejoin in the auditorium in 10 minutes. Thank you. Yes, ma'am.
ఇది కూడా కనెక్ట్ చేసి పెట్టేసి